Good morning everyone, and welcome to my last lecture. So we are going to continue on the topic which we started yesterday, which was about oscillators and 24 hours and chronotherapy. So remember that yesterday we saw several models of oscillators, and then I introduced the Circadian timing system. Which is that, maybe you remember that we said that the whole body has different oscillators, and this induces oscillations in the physiology of the body, and including the drug response. So yesterday I showed you that in mouse experiments, just changing the time of administration of the drug can drastically change the toxicity of the drug, and this was the mice. So a few slides here in humans, because just to show you that this is true in humans. So here I'm describing two clinical trials, the patients who were included are patients who had colorectal cancer, so cancer of the intestine, and they received either standard chemo or chemotherapy, but chronomodulated following this scheme using this device. So more precisely, the device is basically at the bed of the patient and is connected here, and the drugs will go directly into the blood of the patient, and then they will receive the drugs according to the profiles which are described here, which are programmed into the pump. So they receive three drugs. Irinotecan, which is also called CPT11, so the green one. Maybe I will just write the next, because this is going to be throughout the lecture. So CPT11 is irinotecan. So this is the first one. This is given in the morning. The peak is at 5 in the morning. Then they receive oxaliplatin, which is the drug that we have already seen yesterday, oxaliplatin. And the last one, the red one, is 5-fluorouracil. So all this, those three drugs, is the regimen that shows the best efficacy for colorectal cancer. Sometimes in the literature you see this as quite nuts. That's the name of the regimen. So in standard clinics, the patient will receive this at different times. They don't really care about the timing. But here, for the patients who did receive chronotherapy, they received it like this. So irinotecan once at the beginning, with a peak at 5 in the morning, then 4 in the afternoon for oxaliplatin, and 4 in the morning for 5-HO. And the shape is just to avoid toxicity. So I mean, it's not a peak, it's not a bolus, because otherwise it would be too toxic for the patient. Okay, so if you do that, so if you compare chemotherapy to that with standard, what you get is that basically you have a reduction, so you have a decrease in the incidence of severe toxicity. So the patients who receive that, in average, develop less toxicity, so less side effects, and this also translated into less hospitalization. Okay? So people who were back home did not go to the hospital for toxic events. And in terms of numbers, those are the numbers, and now it's also important to understand that there are large sex differences between men and women. So here, this is just the incidence, the percentage of patients who did develop severe side effects. So as you can see, women actually developed more side effects than men in total. Taking all courses of drug. And if you look at the panel B here, the x-axis is you have to see this as the drug administration time. The 0 hour is exactly the scheme I showed you here. So, okay, so 0, when I say 0, I mean 5 in the morning, 4 in the afternoon, 4 in the morning. And when I say here, for instance, -3, I take everything and I shift by -3, okay? I shift the whole thing by -3. So basically, if I do that, I will, by plus and minus 12 hours, I will cover my 24 hours. And basically what you get is that, so you have here, the solid curve is for men, and the dotted line is for women. And as you can see, so we are looking for the minimum, so the optimal timing will be the minimum, this is what we want. And as you can see, for men, 0 is okay. So the scheme that has been used in the clinics is okay. This is more or less the minimum. There is another one here of 96 that sounds to be good. On the opposite, for the woman, the 0 is not the best one. Sorry, 0 is not the best one. And actually, plus 6 hours would have been better. So now we can start understanding that basically the optimal timing will actually depend on the patient. And in particular here, we demonstrate that it depends on the sex of the patient, whether you are a man or a woman. And just to convince you a bit more, I will show you a second example before going to the math. So this is a more recent trial in which basically now everybody has chronotherapy with the same form, okay? Same administration form. And now there are 6 groups of patients, and the difference between the groups is going to be the timing of the first drug, irinotecan, which is shifted in order to get 6 times, which are written here, 6 times of administration. So we are talking about the same type of cancer, colorectal cancer, and the 2 other drugs are given exactly as in the scheme I just showed you, except that here you have 5 fluorescent first, 4 in the morning, and then oxaliplatin 4 in the afternoon, and then you repeat that for 4 days. Okay, and then here are the numbers for men and women. So as you can see, those are low numbers, but actually this is what we get in general in patients. And those are the results. So basically here, A and B are for men and women, and we are looking at the percentage of patients who develop severe toxicities for 2 different toxicities, so the 2 colors are 2 different toxicities. Here we are looking at the time of administration of irinotecan on the x-axis. So what you are looking for again here is for the minimum of this curve, because this is where the least incidence or the smallest incidence of toxicity is. So for man, you can see that it's basically in the morning. On the opposite for women, the minimum is around here, so which is the afternoon. And then if you do that for, or if you draw this for all possible toxicities, because here this is only 2, you can get this type of graph. You can see this on the clock again, and basically each hour will be 1 toxicity, and we point at the time of minimum of this curve. Okay? So for instance, if I take this red curve, it will point at this time, so this is this one, and the length of the arrow will just be the amplitude. So if here my curve has a high amplitude, for instance this red one, then I will have a high amplitude here. And every red-ish color are the women, every blue-ish color are the men, and as you can see we have a very nice alignment. All the men points around here, which is around 9 o'clock, and all the women, the toxicity for the woman point around here, which is around 3 o'clock. So as you can see, we have a consistent 5-hour shift between the optimal timing for men and for women. So, okay, so that's it for the introduction. So the idea now is to say what can we do for the clinicians. We know this, we don't really know why, and this at the moment it's really like a current question, or why do we have this type of pattern? And so the idea was to use mathematical modeling and systems analogy to try to help the clinicians to understand that. So because the problem is very complicated, the idea was to go first at the cell level. So again, we are going back to the cell culture and not to the whole organism. So what I'm going to show now is studying in the cell culture. So I put a photo of the cell culture. This is what you see in the microscope. So here you have thousands of cells. And the idea is to use the PKPD modeling to try to investigate the determinants of these, we call them chronotoxicity rhythms, and also chronoefficacy. Toxicity is activity on healthy cells, and efficacy is activity on tumor cells. So we are going to focus on irinotecan, which is the first drug here, the one that is given the first in the scheme. And we are going to focus on one particular cell line, which are CAP2, widely used for the stomach and so they are colorectal cancer cell line, so they are actually very important to focus. And then remember that yesterday what we saw is that each cell has a clock. Okay, remember that. The only problem is that basically in culture, the cells they are synchronized with each other. So you need to do something to synchronize them, and this is what we have done. So basically, you take your petri dish, you synchronize cells, and then they will all run in synchrony. Now basically what you are going to see is this. So we are looking at basically the x-axis is the circadian time, 0 is the synchronization on cells. Okay, you synchronize the cells, and then on the y-axis you have the amount of some mRNAs from some clock genes or genes which are involved in the pharmacology of the drug. The clock genes are around here. Maybe you can recognize this one, BMAL1. And the blue curve shows the normal condition, and as you can see, we can actually here measure the clock, and we can see that the clock is ticking, because here we do have some oscillations. On the opposite, when you turn off the clock, so this was like control, so you can turn off the clock by just removing BMAL1, so one of the clock genes that will completely abolish the clock. Then indeed, you do not have any, so this is the red line, you do not have any circadian rhythms showing and proving that the oscillations do come from this oscillator. Because in biology, there are kinds of things happening in this. Okay, so now we have these to describe the drug pharmacology, so irinotecan pharmacology, and to model it with ordinary differential equations. So I'm going to have like 4 slides on the explanation of drug pharmacology, because otherwise we're not going to understand anything. So CPT11 is a prodrug that has to be activated into SN38. So SN38 is really like the important guy, that's the active metabolite, that's the one which is going to create inflammation in the cell. So that's a really important one. And this first reaction is actually catalyzed by an enzyme called carboxylsterase. So this is an enzymatic reaction. So what are we going to use for modeling that? What are we seeing for enzymatic reactions? Do you want to use the law of attraction to model the reactions with enzymes? What was the other law that we actually saw? So there was the law of mass action, the second one was what? Anybody, please. The one with the quasi-state assumption. Nobody? Really? Okay, so it's Michaelis-Menten. I'm sure some of you know, right? Okay. So basically when you have an enzyme that is helping the reaction and that is not concerned about the reaction, use the Michaelis-Menten kinetics. Okay? So first step is activation, an activation step. The second one is an inactivation step. SN38 is deactivated into SN38G. So just to sum up, this one CPT11 is not really active, this one SN38 is super active, and this one SN38G is not active at all. Okay? So in the case that would be like sync compartment. And the inactivation step is also catalyzed by one enzyme which is called VGT1. So what are we going to use for modeling that? second one, again, mechanism, exactly. Okay, now let's go a bit more complex. Here this is the cell, and all three species, so they enter the cells passively. So here we are going to use the first flow. On the opposite, the efflux is mediated by some transporters. So these guys here, which are called the ABC transporters, they are pumps that will pump the drug from the inside to the outside. So you can see this as a defense mechanism. When the cells sense something that should not be around, it pumps the drug outside of the cells. So it's called efflux pump. Efflux, I should maybe write that, because you might still be in the literature. And here is going to be the ABC transporter. Okay, so now this is all PK. Okay, now PK is done. We know exactly what the body does to the drug. Now we're going to look at PD, so pharmacodynamics, what the drug does to the body. So what does it do? So here, CBT11 is called an inhibitor of topoisomerase 1. So here, it's going to be intense in biology, but try to stick with me. That's the last slide of the discussion. So topoisomerase 1 is the blue thing that you can see here. And the idea is that topoisomerase 1 is here to help the DNA relax, because sometimes the DNA can be super cold, so the tension in the strands is too high. And basically what the topoisomerase will do is that it will cut one strand of the DNA, then allow the strand to relax, to turn around, and then reconnect the strand. And where the drug attacks the protein is exactly at this point. Basically, when the topoisomerase is on the DNA, the drug will attach topoisomerase 1 and will prevent the strand from reconnecting. So basically, the drug comes here, and then completely stop and prevent these steps to occur. So meaning that on the DNA, you have breaks, single-stranded breaks, with the top one and the drug antibodies. So if we summarize all this, or model all this, so on the left, this is something we have already seen, right? So you have CBT11 that is activated into SN38, so the important guy, okay, you have to remember one, remember SN38, and then you have the deactivation into SN38G. Then the three species here are transported by efflux transporters, but they can also enter the cells passively. Then those two guys, but especially this one, we create something that we call reversible complexes. So those are the complexes on the DNA with top one and the drug. And actually these complexes then will become some much more damageable complexes, which here we call irreversible complexes. Because actually those ones can actually spontaneously dissociate. And those last complexes may trigger apoptosis, so cell death. So I'm going to give you one example for the equations, but I'm sure that you can derive the equations by yourself now that you know everything about law of transaction and mechanics. So this is the equation for CBT11 inside. So this is this guy, the one in orange. Here. So as you can see, and this is not even the complete one, this is just the PK. No, wait, I think we don't actually model that, because we know that this one is not really important as compared to that one. Anyway, so CBT11 here. So this first term is for up CBT. So this is this arrow, the CBT11 enter the cells, so this is a positive term. Then you have two negative terms. The first one here is the influx. So it's this arrow. So we have also modeled this as a mechanism enzyme. As you can see here, we have the B0. So this ABC-CBT is the blue pump that you can see. And we treat this as an enzyme, as if it was an enzyme, because it's actually not consumed by the reaction. So here we have the parameter V0 times the enzyme times the substrate divided by K plus the substrate. That's mechanism term. And the last term is the activation term. So the one of the conversion from CPT11 to SN38. And again, this is a mechanism term. And the enzyme here is CES. Okay, so this is what you can do if you just model the mathematical model. But now remember that we are interested in 24-hour rhythms, in circadian rhythms. Do you have questions? Okay, but you can ask questions. Okay, so now we have the typical PKPD model, but what we want to add on is the circadian rhythms. So the way we are going to do that is to go back to the biology and say, wait, we know in the literature, and actually in our experiments, that there are circadian rhythms in few variables of our model. So wherever I put a clock, it means that actually there was a demonstrated rhythm, meaning that basically the amount is not constant over time, but it's cycling over time. So we need to represent that in the model, and so there are basically two ways. So either you use an oscillator, there is two possibilities to represent this in the mathematical model. Either you use an oscillator just like we have seen yesterday, so we use the dynamical system that we have spontaneous oscillations if you choose the correct parameters to have a cycle. This is one possibility, which is the most complex. A simpler one, if you don't have information, is just to force oscillation using a simple cosine function. And this is what we have done. So basically now, for instance, if I write this guy, CES, this is not a constant anymore. And now CES is going to be a function of time. And we just follow a cosine function. So we have a simple average value plus some cosine term. A is the amplitude, capital T is the period, and phi is the phase. Okay? So that's the mean. Sometimes we call it the measure. Sometimes you can see this in the literature. This is the amplitude. This is the period. And this is the phase. Okay, I'm sure you are all familiar with this, so this is just a reminder. And just again a reminder, if you write this, you will get the phase here. That's the time of the peak. But if you write this like this, the mean would be here. Actually, the mean here is 0. The amplitude would be that. So the difference between this and this should be the amplitude. And what else? And the period is that. Okay, so what we do is we use that for any protein that has a circadian rhythm. So here, as you can see, we have 4 plots. So we have 4 cosine. And what we have basically is that for each cosine we have 3 parameters, M, A, phi. So the period T we have already estimated from the first data I showed you, the mRNA data. So when I say I'm going to estimate my parameters, I include this A and this M, this A and this phi, and this phi into my parameters to estimate. So okay, the model is done. Okay, the model design is done. I have I would say a regular PKPD model that is forced to oscillate by some forcing functions which have the shape of oscillators, not oscillators, they are cosine functions. So the next step, of course, after model design, we go to parameter estimation. So I will probably not go into the details, but I will just show you the data, just to show you that we do have data. So we started by the PK from the kinetics. What you can do is measure basically every mathematical variable that you have in your model, you can measure it. So here, this is extracellular CPT11, intracellular CPT11, extracellular SN38, intracellular SN38. And the three colors correspond to three types of administration of the drug, so of this guy here. And basically what you can see, if you focus maybe on this one, which is the most important because this is the active metabolite inside the cells, so this is what is going to be important for the efficacy. What you can see is that basically we have already large differences between the three types of administration regarding the accumulation of the drug, well, of the metabolite into the cells. If you just focus on the blue and on the red, as you can see here it's almost double, it's actually almost here you have 0.04 and here you have 0.08. So basically just changing the time of administration will double the quantity of active drug that will actually accumulate into your cells. So it's almost as if you are changing the dose in a way, because you are doubling the active metabolite. And just to convince you that this is not something, that this phenomena actually arises from the clock, because it could be something else, okay? It could be something else not related to the clock. What we did is that we performed the same experiment, but this time by removing, when we are removing one clock gene. So basically, the right column here is when you remove one clock gene, which is DMARA. And if you focus on that, you will see that now all the points here are superimposed. And so meaning that for any time of administration, you have the same amount of active metabolite inside your cell. So meaning that this really demonstrates that this phenomena comes from the circadian effect. Okay, that's PK. Let's go to PD, pharmacodynamics. So we have measured two things. So we have measured here the quantity of complexes, and here the apoptosis, the cell death. So here the three bars are actually the three experimental results. Sorry. Again, the three colors correspond to three times of administration. So remember, 0 is the time when you circulate the cells. So here you have 2, 14, and 20. So that's 2 hours after cell synchronization, 14 hours, 20 hours. And the line here is the mathematical model best fit, including all data I showed you, okay? Not just this one. This is the reason why you don't really have something really fitting with the 3 points. It's because we also integrate the PK data that you have just seen, and also actually the apoptosis data here. And again, the left column is in normal conditions, and the right column is whenever you turn off the clock. And as you can see again, so the amount of reversible complexes and the amount of cells which are dying are highly different when you change the time of administration. If you look at the death, for instance here, if you give the drug around, so we call it CT for the circadian time, so around CT2, here you have about 15% of the cells which are dying. On the opposite, if you give it at CD20, you have more than 70% of cells which are dying. So that's actually a huge difference. Okay, so until now, up to now, we have a mathematical model that is now calibrated. Okay, we have the model design, parameter estimation. So now let's go to parameter importance or sensitivity. So for this, we have used the Sobol. I'm not going to go through this, but maybe you are familiar with that. So we have used the Sobol-based approach to compute the sensitivity of the parameters. And here, the question that we ask is which of the three zones, the clock, which of the clock here, which of the clocks here, or 5 of them, are responsible for the results we see here in the apoptosis, or maybe if I have it, let me just go back here. I want to understand this basically. I want to understand why I have this difference in cell death. I mean, which protein is responsible, or which proteins are responsible for this particular pattern. So what I do is that I basically take all my parameters of all my model, including the ones that I actually described here. So here remember that you have for carboxylic but for any 5 clocks, you have M, A, and phi, so amplitude and phase. And then you put all this in your sensitivity analysis, and what you get the result is this. So here we are looking at, so each column is one parameter of the model, and here we are looking at the sensitivity of the parameter. So if it's high, it means that it has a high influence on the chronotoxicity rhythm. And basically the first one, let's say the first, I would say the first, or almost the first, except this one which is exposure duration, which is a bit trivial. Among the 5 first one, the 4 here actually relates to UGT1A1 and carboxylase bases, which are those two guys here, okay? This guy and this guy, so the activation enzyme and deactivation enzyme. And here basically the phase of the deactivation enzyme and the phase of activation enzymes, so this parameter here in the forcing functions, are the most important parameters to predict the chronotoxicity rhythm, followed by the amplitude of the same proteins, so of the same forcing function. So if you translate this in terms of biological meaning, it means that basically if you wanted to predict the optimal timing, so the time where the cells are not dying, because they say that you don't want them to die, then you will have to track those two proteins, carboxylase bases and UGT1A1. Look at, if you cut here, you just want to look at their phase, for instance, and then if you have the phase, you are very likely to be able to predict the optimal timing using this mathematical model. Okay, so basically at this point here, we have a model, it's calibrated. So the next step that they want to do is to go through therapeutic optimization. So here, we are not going to optimize drug combinations, but we are going to try to optimize timing of administration. Yeah, because here we are looking at timing. So I'm going to define two cell populations in a different way as compared to what we have done already. Because we know that in tumors, the clock may be disrupted. We know that the tumor tissues may not have proper circadian rhythms. So either because the cancer cells do not have a clock, maybe the clock could be broken, the clock could be hijacked. So and of course the clock may not be synchronized throughout the tumor. So if you look at the clock at the level of the cell population, of the cancer cell population, even if you have oscillators like this, which are present but which are desynchronized, if you just look at the average, you will get a flat curve. Okay? If you take your cells, 1 million of cells, they oscillate but with different phases, periods and amplitude, at the end you will just get a straight line. So this is what I assumed. So for the cancer cells here, we are assuming that basically they are not synchronized in a way that basically all the cells are now freely running, so we don't know what they do. They may have results, but they may not have, they are not synchronized. And overall, my pattern in my mathematical model for the proteins are going to be flat. Okay, for carboxylase, for CES, for UGT, for ATP transporters, everything will be flat. On the opposite, here, for the healthy cells, we keep what we already have. We have a nice cosine, which means that the healthy cells are nicely synchronized, and they do have a clock that is working. So, very well synchronized healthy cells, the one I want to keep. Desynchronized cancer cells, the one I want to kill. Okay? Okay, so let's have a look first, not even making any optimization. Let's draw the model simulation for some exposure to irinotecan. And so this basically could refer as the strategy one, which is just maximizing efficacy. So here we only consider the cancer cells, so they don't have any results. So this is flat. Your mathematical model don't really have the forcing function, and all enzymes will have flat pattern. And here we are looking at the DNA image on the cancer cells with respect to exposure duration and with respect to cumulative dose. So of course, when you increase the dose, you increase as well the image. And as you can see, there So here you expose the dose to a fixed, you expose the cells to a fixed dose, and basically you expose during some time. So here you have the time of administration. I want to put it, I will put it tau. Okay, so that's the time of exposure duration. And this one, the area here will be the cumulative dose. Which is here you put some CBT0, some initial dose. This is what I call cumulative dose. So meaning that basically if I reduce, so for a fixed cumulative dose, for a fixed area, if I reduce the exposure duration, I will increase that. Okay, does that make sense to you? If basically if you fix the area, you can just play between the two lengths of the rectangle. So this is why you get this pattern here. So the tau here is just this variable, exposure duration. So what it means, I mean, what this result means, it means that basically if you dilute your drug, you will get less effect, which is a bit intuitive as well, right? You know, if you give a bolus, so if I draw something around here for a fixed cumulative dose, I may get something like a bolus for a small tau, and if I lengthen tau, I will have something like that. Okay, so the cells will be exposed to a smaller amount of drug in a longer time as compared to this one with a small tau. And this is what the results are saying is that basically it's better to have this if you want to maximize your efficacy. So this is something that is intuitive, right? I mean, if you want to give, you need to give plenty of drug in a short amount of time in order to be very efficient. If you dilute, at least in the model, if you dilute your effect by giving a small amount of drug in a longer period of time, you will get a smaller effect. But to be honest, this is not always true in all biological models, biological systems. But in that case, the model predicts that. Okay, so now the idea is to maximize efficacy. So we want the maximum of this curve, and as you can see, we have a line of maximum here. That is to say that you have to expose your cells during during 2 hours to 8 hours, and this duration will increase with the dose. So around here we are around 2 hours, and around here we are around 8 hours. That's the optimal way to kill your cells. So this is not really optimization. You can see this is just running your model and looking at your simulations. So now, again, not optimization here, just looking at the model, we are going to look at the normal cells only. So this is strategy 2. Strategy 2 will only focus on the healthy cells. So remember, the healthy cells are now oscillating, they have a clock, and we want to minimize the toxicity on them. So again, the z-axis here is the effect of the drug. Here the y-axis is the duration of the exposure. But here now, wait, be careful because here this axis, the x-axis is not the same. So now we have the time of exposure, CT for Circadian time. And as you can see, basically, if we are looking for minimum value of toxicity, we are looking for the dips for the holes that you can see around here and around here. So this is where the healthy cells are basically sheltered from the drug effect, just looking at the circadian rhythms basically. And you have some plateau here, in which you have the maximum side effect, which is where you don't want to go at all. And as you can see here, the picture is not very nice for us because you have plenty of red. I mean, if you just look at this part, you have plenty of red if you go along that, and you have only a small amount or a small interval of blue. So it's basically easy to do plenty of damage, to have plenty of side effects if you are not careful with the time of administration that you choose. And so those dips here correspond to CT2 and CT3, so circadian time of administration between 2 and 3. So this is what we have here. And as expected, if you expose the cells to some drug for more than 24 hours, the effect of the timing will vanish. Because imagine that if you provide the drug for 2 days, let's say 48 hours in a row, you don't really have a circadian effect anymore because the drug is going to be in the cells along the whole period, along the whole circadian period. Okay, so now we want to combine both, okay? This is where optimization goes. So we want to maximize the efficacy on the cancer cells with the constraint of side effects. We don't want too much side effect. And this is what we have done. So that's the strategy 3, maximizing efficacy under a constraint of not having too much toxicity. So we don't really have an idea about the toxicity threshold that we can keep, so we have just explored different values. So the toxicity, if you just focus on B, the toxicity threshold is here in the y-axis. So you can think about, okay, I have one patient, well, he's frail, I'm not sure about whether he can actually handle things, so I'm going to put a low threshold. On the opposite, I have a young patient who seems to be very nice, I mean, very healthy, and in that case, I will put a higher threshold. So you put the threshold, and then the green bar will give you the optimization. So again, we program this cost function, and then we optimize that using CIMPA and ES. And what we get is this. Basically, here you have the cumulative dose that is predicted to be optimized by the model. And here you have so on the x-axis, this is the time of administration, and here you have also the duration, the optimal duration of administration. And basically, as you can see, the threshold will not change much, in the sense that it will change the dose, but it will not change the window here, which is again around, so 2 and 3 will be around here, so it's around 2 and 3, but then it's shifted a bit towards 8. And the duration is also not changing too much. As you can see, the duration is like about, it's between 1 hour 30, no sorry, it's between 4 and 9 hours. And the time window that is targeting is also more or less the same. So what it's saying here is that basically for any toxicity threshold, you will have to target the same window, so the time is the same timing, and it's just the dose that may vary. And so maybe what is interesting is that if you look at that, I mean, if you look at profiles, so here we are looking basically at the forcing functions, which are the best fits, so we take them, those are the ones from the parameter estimation. And here, so remember that from the parameter sensitivity analysis, we have seen that there are two proteins which are actually very important. Carboxylate kinases, the green one, and UGT, the blue one. And basically, if you focus on the green one, you can see that here you are basically at the source of the curve. You are at the minimum values of the curve, showing that basically what the model is predicting is that you have to target the time when the activation is the lowest. Okay? And basically, so you may think, well, that's a bit strange, I have to target the time when I don't have my drug that is working. But then you have to go back to the definition of the healthy cells and the unhealthy cells. The healthy cells are the ones that we want to shelter from the effect of the drug, and those are the ones who has secondary results. The other ones, the cancer cells, they absolutely don't care about the timing. So basically what this is telling you is that you should choose the time of least toxicity, and because the tumor cells don't care about the timing, they will be killed in the same way anyway. Okay, so that's it for this part. So just to wrap up a bit of PERC, so we have seen an in vitro study in cell culture. We have designed a PK/PD model of irinotecan pharmacology, and we have concluded through sensitivity analysis, that basically two proteins, so the activation enzymes, ES, and the deactivation enzyme, UGT1A1, were the one driving the difference in toxicity that you see when you vary the timing of administration. And basically, this is probably the summary of what we have done. And we have just extended a bit what I showed the first step, but we do follow the same steps. Okay, so before going to the last part, do you have questions about that? We have optimized this using an optimization algorithm. So you ask the question, I want to maximize my efficacy under some constraints of toxicity, of tolerability, and then this is the results of your optimization. So I am not deciding this. This is the algorithm that will tell you if you want to maximize the efficacy and minimize toxicity. Voilà. Under constraint of toxicity. This is going to be the optimum. This is a numerical result. I don't decide this, the algorithm computes this for me. Any other questions?
Is this applied already in the medical?
I can't hear you.
I'm just curious. I'm just curious if this procedure, the drug administration, is already applied, because the setup is more or less controlled, but in the circadian, the experimental setup. And in the, for example, if you say in humans, it's affected by different factors like pattern, the light exposure, your stress. I'm just curious if it is applied and how much does it really affect.
Thank you for the question. That's a really interesting question. So, and the answer is yes, it's already applied. So there are several fields in which it has been applied for a long time. So for instance, if you think about arthritis or any inflammatory disease, so there has been drugs which are specifically developed to be released into the body at specific times of the day. So for anti-inflammatory drugs, we are targeting the second part of the night, which is where the cytokines will rise. And so this has been around for a few years. For cancer, so this was applied actually in Paris, in one of the hospitals, but it was more like to understand the results, but they were clinical trials, they were not everyday clinics. And basically at the moment we are in terms of impact, so it really depends on the drug and really depends on the pathology, but mostly on the drug. And basically the highest impact we are seeing now is for one cancer strategy, which is immunotherapy, immune checkpoint inhibitors, and you can see that the survival is actually quite longer in patients who receive the drug in the early morning as compared to any time actually in the day. And if you basically, so the worst time is likely to be beginning of the afternoon, best time is early morning, and basically you have an hazard ratio of the survival of about 4.8, so meaning that you have 4.8 more time more risk to die when you are treated in the afternoon as compared to the morning. So it could be really large. I mean, as you can see, I mean, this one is an example that the impact is really large. And I also showed some, maybe I can go back, but you may see that, I will just go back to this one. So, especially for women, you can see that if you just shift by a few hours your drug administration, here you have 0% of the patients who have some side effect, and here you have about 100% of the patients who actually, the women who actually develop side effects. So the impact could be really large, but of course I'm showing this for drugs for which it matters, but there are other drugs for which it doesn't matter. So you need to go drug by drug. Okay, so if there are no other questions, we will go to the last part of my talk, and actually of my lecture. Okay, so remember that I said that to actually perform chronotherapy in the clinics, we need a pump that will help you administer the drug at any time you want throughout the 24 hours span. Because of course, if the best timing is at 4 in the morning, for instance for 5 fluoroacetate, it was at 4 in the morning, you can actually wake up the patient to administer the drug, and you don't want the nurse to work at night neither, because that's not good for him. So we really want administration devices which are accurate. And this is the topic of the next section. And basically, I'm going to talk about one trial, which was a bit specific. So it's called the Optilip trial. So again, it's in colorectal cancer, and it counts only 11 patients. It was really a small trial. And the particularity of this trial is that the pump was not connected directly into the blood, but was connected in the liver, directly into the liver of the patient, so around here. And this is because the part of the tumor was there. So the idea is that basically you administer the drug where the tumor is. Okay? So the patient received actually the same scheme as the one we have already seen. So again, it's irinotecan. I think it's okay. Irinotecan. Then the peak here is a glucose rinse. You want to rinse your. So here you have to imagine this is the tube. Okay. You want to rinse your tube before giving the second drug here, because you don't want the two drug solutions to mix. So you administer irinotecan, you rinse, oxaliplatin, you rinse, 5-HU, you rinse, etc. Okay? And here you have the days like night, day, and so our colleagues came to us and said, wait, we have data in the blood of those patients. We have measured the drug, the 3 drugs, in the blood of those patients, not the liver. Okay? And can you model this using compartment models? And then we said, yes, of course we can. So then we went to typical compartment models, just like the ones I showed on the first day. So we did one per drug, because the drugs are not the same. I'm showing the one for fluoroacetate and for oxaliplatin. The one for irinotecan is a bit more complicated, because as you can see, you have the active metabolite and sortate. So basically, we have 3. So let's talk about the fluoroacetate, this one. We have 3 compartments, so 3 state variables, which represent the blood, any other organs, and the liver. So, of course, here we need to introduce the liver compartment, because this is where the drug is going to be added to. Okay? So you have a pump here at the top that will deliver some quantity of drug in the liver. Then the drug can be directly cleared from the liver through the bile, or it can go into the blood, and actually the drug can also go back to the liver. Then you have some clearance here in the blood, which is basically the renal clearance, so the clearance that goes to the kidneys. And then you have here another compartment which has the volume of the rest of the body. So as you can see, this compartment, or as you can imagine, this compartment is going to have a very large volume, as compared to the other ones. For oxaliplatin, it's a bit more complex because the binding of the drug with the circulating protein is very important. So anytime you have F, it means free. Every time you have B, it means down. And anytime basically the drug is bound to the protein, it's completely attached and it's lost. So basically all this, well, it's almost lost. Basically all this means that the drug is going to be stuck in some compartment and the drug is going to become completely useless like in this. In terms of parameters, B is higher, much greater than U. So the binding, so going in that direction, the binding is occurring at a higher rate, at a much higher rate than the unbinding. And then for the rest of the model, it's the same, except that we know that there is no clearance here because the drug didn't go to the bind. Okay? Okay, so we thought it was simple. We just applied this to the data, and this is what we got. So here we have the data for the 11 patients. 10. So for this, I'm showing the data for oxaliplatin, okay? So for the second drug here, oxaliplatin. And as you can see, for some of them this is working. So for instance, this one, patient 6, we are happy. For other ones, well, let's see. What about this point? What about that point? Actually, I'm going, yeah. What about this? What about this? I mean, it looks strange. And it repeats itself. I mean, for several patients, we have this. Sounds not really, I mean, it's not captured by the model. Something is happening here. And something else that is not really well working is that for the second point, for some of the patients here, actually the model is not capturing the delay because we do have a delay. For instance, for this one, the concentration of the drug stays below 0 or close to 0, sorry, during several hours. So we said, well, there is a problem in the model, so at least the model does not represent the reality. And we said probably it's because of the pump, because we haven't actually described the pump in the model. So we said that let's actually make a mathematical model of the administration pump and the tube from the pump to the patient. And then once we have those profiles, those administration profiles, let's put them into the PK models. So this is what we have done. So we have done one-dimension transport equations. So this is 1D, meaning that basically we assume that the tube between the patient okay, so basically for this PD, we assume that the domain is just one dimension. At 0, you get the pump here. And at capital L, you're going to have the patient. Okay? Here you have capital L, and that's the patient. And basically we know what we have programmed here. Okay? So this is something that was programmed by the humans. And what we want to predict is what the patient sees. What is actually delivered into the blood of the patient. So so here you have the X, well that's the X variable. And then you have a transport equation here. That depends on the velocity of the fluid. And basically here what you can see are simulations. So this is the distance, so 0 here you have the pump, and here you have the patient, and here you have the time, the normal time. And as you can see, that at the beginning, so here you have the scale, so blue is 0, and then you have red. So over time, basically, so at the beginning there is blue everywhere, so there is no drug anyway. Then the drug is administered using some bell-shaped profile. So you go here, and then basically the drug is actually going further and further and further, and at some point you have the drug all through the tube. And so this is this regimen here, and and then you continue, and then you rinse with the glucose. Remember that you have a rinse. So here the switch between the red and the blue is when you rinse with the glucose. Okay? So you go back from drug to no drug because you just rinse. So it is an intervention by the clinic. And just looking at this, just using this PDE, what you can do is you can actually compute the quantity of drug that you can see here at X equal capital L, because this is what you want to know, what is the profile of the administration in the blood of the patient. So this is what we got. So you can actually derive here, that's the drug infusion rate dt, that can just be computed as so SA is SA is the surface, because we know the tube. Okay, we know the tube here, and SA will just be this unitary surface. You can see that. So we know the diameter of the tube, and so you just want to compute that, and basically what you get is all the following profiles here. Which I'm going to explain. So basically, you give or you provide maybe I'm not sure you can see, you provide the blue one. So here you have the blue curve here. This is what you provide. And actually the orange is what you get. So here here you have a discrepancy between the two curves here and there. So here basically you have like a delay between the blue and the orange, and here you have a peak which is not present in what you is intended. So basically the blue is what the clinicians want, what they program into the pump. The orange is what we predict with the PD. Okay? Can you see the difference? Okay. And basically it's the same for all three drugs. We have a delay here, which is actually larger or longer for oxaliplatin as compared to the irinotecan. And then we have here a peak that is occurring in terms of drug administration. And here on the right column, we have actually depicted the total drug. So this is in percent, so the total drug that is delivered to the patient. So at the end, he or she will get 100% of the dose. And basically, what we can see is that if you look at oxaliplatin here, when you look at the orange curve, you can see that the dose actually starts later, it starts being non-zero later, and then you suddenly have here a step which corresponds to that peak. So what does it mean? I mean, basically, it could have been maybe guessed, but here that's really the mathematical modeling that gives me some numbers or some quantification. So if I focus on that and accuracy, it means that here I start activating my pump, so my pump is pumping, but wait, my drug is in the pump at the beginning of the tube, so I am at 0. And then I need some time for the drug to go from 0 to capital L, because I need the drug solution to push the solution that is already in the tube, which has 0 drug in it. Okay? So this is this delay. Here, this is the time for which that needs the drug to go from 0 to capital L. Once the drug reaches the patient, we are here, and then the drug will start accumulate, or will start being delivered, and you actually get the same profile as intended. And then, okay, wait, here there is a rinse, okay, you rinse with the glucose. But when you rinse, the solution that was in the tube contains drugs. And basically what you have here is a shot, like a bolus, like a peak of administration, just because you push the drug solution using your rinse towards the patient. Okay? So this is something that was not expected, or let's say not taken into account by the clinicians. And this is something that, of course, has an impact on the PK. So now that we have this mathematical model, you can just actually use this dt here, this rate of administration, as an input for your OD model and for Pathom model. Instead of the blue, basically, instead of the blue here, you take the orange. And now you resimulate, okay? So now you go to, so here I am showing oxaliplatin again, and basically what you get is this. So now the blue curves are the curves that you get with the same mathematical model, the same parameters actually, for the ODs. But now the administration is not a simple bell shape. It's a bell shape that is delayed at the beginning, basically the orange curve, and then with a peak at the moment of at the time of the glucose rate. And as you can see, you do have delay. Now look at those guys. I mean, before with the first mathematical model, we were unable to show the delay, but now we do capture the delay. And also, as you can see, we do capture the peak here, and here, which corresponds to the rings of the glucose. And for some of them, so for instance for these patients, the peak was not even in the data, because the data was not sampled in a sufficiently precise or accurate manner. And the model actually predicts things which are not in the data. But it does predict, remember that we saw at least those three guys with this particularly high values. Now the model can actually really predict those outliers. Okay? Okay, so basically what we have done, what you can do is one more analysis, because I should have said this maybe earlier. So the parameter estimation, what we have done is we have done one estimation for one patient, because we only have 10 patients. So basically each patient has their own parameter values, okay? Because we know all patients are different. And that's the last thing, I think that's the last thing that we tried. So we actually tried to see whether or not the parameters have a correlation with, so the PK parameters have some correlation with some characteristics of the patient, because remember that for instance the sex could be important. But unfortunately, there were no correlation between, for instance, sex and the PK parameters. And then we also tried to cluster the patients to see whether or not we could have patients with different PK for the three drugs. But it was a bit, as you can see, it was not really convincing. So that's the clustering for the three drugs. We only have 10 patients, but I mean, maybe the only thing we can say is that here we only have one patient here for that maybe an outlier, but that's clearly not, we don't have that patient here. And if you are interested in the parameter values, I plotted them here. And basically, so each line is one patient. So again, so the so three drugs, three drugs. Then one line equal one patient, and here you have the parameters of your departmental model. And just looking at this, it's interesting to see that the variability is localized in some parameters. Okay, so here we are looking at the inter-patient variability. And for instance, if you take that one, the model irinotecan, this parameter is the transport between the blood and the organ. Actually, here, for oxaliplatin, this is the transport rate between the blood and the liver, and here this is the transport rate between the blood and the organ and the blood and the liver, which are supposed to be equal. And as you can see, basically, the variability is clearly in the transport parameters and are not are less present in the clearance parameters. If you look at the capital C here, and there, or except maybe this outlier, you can see that the variability is really located in the transport parameters. So maybe this is where we expect interpatient variability, and if we wanted to choose some parameters to actually set to a unique value for all patients, we'd probably choose the capital C of the clearance and not the transport. Okay, so let's wrap up for this part. So here what we have discovered is that there was a misuse of the administration pump, simply because the clinicians did not think about the tube, the volume of the tube between the pump and the patient. So it takes some time for the tube to be filled with the drug solution, and then when you rinse the tube with the glucose solution, you are going to administer the drug in an unwanted way, because it's not what you want to do. And if you think about chronotherapy, in particular, you are going to administer part of the drug. So maybe I haven't said this, but for oxaliplatin, it was 10% of the dose that was administered in the peak. 10% of the dose, so that's not a small amount, okay? So 10% of the dose was administered during the glucose peak, which do not occur at all at the time where you actually want the drug to be. So for chronotherapy in particular, this is really important to actually have accurate administration devices, and this is the way we can help. So because basically we have asked for a pattern for this, for the PD model, so that this part of the model can be included in the software of the pump to calculate, to compute the actual drug rate that you need to achieve what the clinicians want. Because basically now that we know all this, and now that we know the delay and now that we know the peak, you can change, you can modify your drug rate administration so that you do get what you want in the patient of the drug at the end. Then we have suggested a combination of PDE and ODE model based on the compartment models. And we also actually had some, say, preliminary conclusions, because this is only 10 patients, so this is not bad, but preliminary conclusions about what could be responsible for the inter-patient variability. In this case, it should probably be transport, and it should probably not be the clearance of the drug. Do you have questions on this part? So if not, this is the end of my lecture. I'm a bit early. Do you have questions on anything else? Because since we have time. Yeah.
First of all, I'd like to thank you for the nice presentation for the entire week. I have enjoyed your talk. And I'm quite new to the area of pharmacokinetics and also pharmacodynamics, but I could see through all the presentation about the mathematical models that the PEEs and the ODEs were with the integer derivative. So I'm wondering if, like, because of the complexity of the interaction between the system of human body and the cancer cell, for example, so I'm wondering if it is possible to model by using differential equations with fractional derivatives. Are there any application problems that people are using mathematical model with differential equations that contains fractional derivatives?
Okay, I'm not aware of. So the question is about I'm not aware of such work. Maybe this exists, but I'm not aware. The thing is that, of course, we would prefer to stick with tools that everybody uses, so that we all have comparable results. So I think in terms of differentiation, we stick to PDs and ODs, like the ones I presented. So I guess we need to look in the literature to answer your question, but I'm not aware of such work. I guess you are working on fractional differential. Are you aware?
I'm trying to work on it, but I'm pointing to the Pathom planet. You could say I have zero knowledge.
Okay, any questions from the audience?
For maybe the second or the third slide of this one, for the model, this one. For 5 you have only 3 parts, right? But for oxaliplatin white is not the second one. Please explain.
Thank you. So it depends on the pharmacokinetics of the drug. So here for acid, the binding is happening really quickly. So we actually assume that there is already an equilibrium between the it's not that they're combined. It's more that this is already gone. So basically we don't take this U. And now, if you just have that, this is just a single compartment, something that is never come back, and you don't have to represent that, just like as examples. So for oxaliplatin, this is happening in a less intensive way, so basically this is faster here. So we decided to actually keep this. And another important argument is that for oxaliplatin, we actually have data for that and for this. So whenever you have data, of course you want to use it just to make sure that your model is actually accurate. And this is why we decided to have both. For 5-fluoroacid, we do not have data for the time of the start, of the bound action.
Hello, Dr. Ballesta. I'm just wondering how the pump is attached to the body with the 3 cancer treatments. How is it attached to the patient?
Thank you for the question. So the cancer patients, they are operated because they are going to have plenty of drugs. So they actually have a small surgery, and they have a catheter here that will actually be underneath the skin. And that will be directly connected to the artery, and then you can connect the tube of the pump, and then the pump will be attached here. And you have the pump plus 4 different bags where the drug solutions are for the 3 drugs plus the glucose for the rinse. Yeah, and then the pump will handle the administration and which drug to give at what time.
Thank you very much, Dr. Ballesta, for your very inspiring talk. And this is just on the interaction between the math people and the experimenter in your experience. I'm just curious about their interaction. Is it always the mathematicians who are going to the experimenter? We could do something like this, or is it the experimenter who are presenting always to the math people? Can you help us solve this, or anything could happen in your case?
Thank you for this very interesting question. So I would say that most of the time the biological questions will probably come from the biologists or the clinicians, because they do have a problem, they work on something, and at some point they are stuck. They have a problem, they don't know how to deal with that. For instance, here we saw the PK profiles with these outliers. It was something strange, and then we come to the game and we try to do what we can. So then in that case we go to them. Well, to be honest, there are also, now in my lab, for instance, I also initiate other projects, and basically I coordinate research projects in which there are biologists and clinicians who are doing, as collaborators, the experiments. But the experiments are decided together, so that I do have the correct experiments, or experiments that I need to design my model and calibrate the parameters. So yeah, but most of the time it's really like the mathematicians that need to go and understand the biology. I mean, it's really rare that actually the biologists or clinicians will come and understand the mathematics and specifically ask for OD modeling. This is not happening. So you have to go and try to speak the language of the clinicians and of the biologists, and don't go for them to actually speak the language of mathematics, because this is quite hard, and they don't have time in their busy, and that makes sense because remember the clinicians are dealing with patients who are the most important ones.
any other questions from the participants? Thank you very much for the question.